So we're going to go into the specific effects of insulin, and first on carbohydrates. So there are really a variety of things that get activated, a variety of things that get inhibited. What gets activated is glycogen synthesis and glycolysis, two processes that consume glucose because there's plentiful glucose in the bloodstream. Okay? The reverse processes, glycogenolysis, which produces glucose, and gluconeogenesis, which also produces glucose from smaller molecules, are inhibited. And that's specifically important in the liver, because the liver is the only tissue that has gluconeogenesis. In both liver and muscle, you have a decrease in glycogenolysis, degradation of glycogen. The tissue-specific effect of insulin is this last one. This should be really highlighted, very important. Insulin stimulates glucose uptake in these specific tissues, muscle and adipose tissue, by increasing the number of GLUT4 transporters on the cell surface, in the cell membrane. So more transporters, more glucose taken up into the cell. The effect of insulin on lipid metabolism. Let's start with adipose tissue. You remember, adipose tissue is where we store a lot of tags. The adipocyte is roughly 80% tags. Okay? So adipose tissue, important effects on adipose tissue is first to decrease lipolysis by inhibiting hormone-sensitive lipase. Second is you increase tag synthesis. And how are tags synthesized? That you need fatty acids and you need glycerophosphate. The fatty acids are coming from the dietary tags, from chylomicrons. Lipoprotein lipase breaks them down, picks those fatty acids up into adipose tissue. Then they are esterified to glycerophosphate. Where is the glycerophosphate coming from? It's coming from DHAP from glycolysis. So remember, adipose tissue picks up glucose. It's using glucose for its own energy needs. So it will have an ample supply of DHAP and glycerophosphate as a result. And that's the backbone used to esterify fatty acids from the diet or from de novo synthesis from VLDL, and you increase tag synthesis. Okay, so glucagon is going to have, again, its own set of effects on metabolism. The effects on carbohydrates primarily are to increase glycogen breakdown, glycogenolysis, by activating pKa phosphorylating glycogen phosphorylase kinase, glycogen phosphorylase kinase, phosphorylating glycogen phosphorylase, and that results in glycogenolysis. Okay? Simultaneously, it will inhibit glycogen synthesis. That's always very important. You're activating one thing, inhibiting another. You know, so you're not having both occur simultaneously. And the inhibition of glycogen synthesis is because you phosphorylate glycogen synthase by pKa, which becomes inactive under those conditions. You have an increase in gluconeogenesis. Okay? Both of these are key functions, again, of the liver. These are the key functions of the liver. So think about it. Glucagon is high when glucose in the bloodstream is low. That means you are in a fasting cycle. So the liver's function is going to be completely changed, meaning it has a new function. Its new function is going to be producing glucose. Okay? How do you produce glucose? Through either glycogenolysis or gluconeogenesis. What happens in the with lipids? You're going to use whatever tags that were stored before, either in adipose tissue or the liver, as a source of fatty acids. So you break down tags. Lipolysis will be increased primarily by activating hormone-sensitive lipase. Okay? But I want to kind of point something out. Adipose tissue doesn't have a glucagon receptor. So hormone-sensitive lipase is activated in adipose tissue by epinephrine through the beta-adrenergic receptor. Okay? So it's a unique and important kind of detail to pay attention to. So you have fatty acids are going to be used as a source of energy instead of glucose in the liver, in other tissues, in the muscle, in the heart, in many tissues that can use fatty acids, but not in the brain and not in red blood cells. Okay, these two organ and cells still need glucose as their major source of energy. Now, what's going to happen as well is that the liver will be increasing ketone body synthesis, ketogenesis, because it's using fatty acids as a source of energy, producing a lot of acetyl-CoA, and some of that acetyl-CoA is used in ketogenesis. So note how important the liver is. It's providing glucose, providing ketone bodies, two different forms of energy to other tissues that can use those forms. In terms of gluconeogenesis, what's going to happen, where the building blocks or the precursor molecules for glucose coming from? They're going to come from muscle amino acids. So in the liver, there will be increased uptake of amino acids. Again, muscle protein turnover. It doesn't mean that you're wasting away. It just means normal muscle protein turnover. Breakdown of muscle proteins gives you amino acids. Amino acids released in the bloodstream, picked up by the liver. So this is amino acids coming primarily from turnover of muscle proteins. Okay? And they are going to provide the carbon skeletons for gluconeogenesis in the form, we talked about one of them, alanine can be released, goes to the liver, becomes pyruvate. Pyruvate can be used to make glucose. So here's a summary of the effects of glucagon. Pretty straightforward. Without going through all of them, here's mainly effects on the liver. Activation glycogenolysis, inhibition of glycogenesis, activation of gluconeogenesis. This one here, note, no effects on muscle. Really, no effects on adipose tissue. This is an effect of epinephrine on the beta-adrenergic receptor that in turn activates adenylate cyclase in adipose tissue, producing cyclic A, activating pKa phosphorylates hormone-sensitive lipase that breaks down the tags in adipose tissue and make those fatty acids available to the rest of the body. Okay, so that's why it has a different color. This is due to epinephrine. If you like this video, please subscribe. Thank you.